Hello. Today we'll talk about response to antigen, and I will see in more detail how the immune system can respond to an antigen through some signaling receptors on the surface of the immune cells, as well as we will see the interaction between the TCR receptor and the antigen-presenting cells, and we'll see more details in this lecture. So let's go through the fate of the antigen, either a microorganism or a protein or exogenous protein, of course, or even a vaccine that got internalized inside the body. And we'll see how the humoral and cell-mediated immunity will coordinate to perform its function. So if the antigen actually entry was intravenous, so phagocytosis and pinocytosis occur in this plane. And if the entry is other than intravenous, so it will be recruited to the lymph node and will be processed by the antigen-presenting cells, monocyte, macrophage, and dendritic cells. So actually, we have two different antigen categories, either an intracellular organisms or a protein, exogenous protein. So let's talk first about the intracellular organisms. So if an intracellular organism, the antigen from this intracellular organism will be synthesized in antigen-presenting cell cytoplasm and processed to peptides. And soon after that, the peptides, together with class 1 MHC complex, will be expressed on the membrane of the antigen-presenting cells. Soon after that, the CD8 TCR receptor on the T cells which actually the TC cells will bind to this complex, the peptide class 1 MHC complex on the surface of antigen-presenting cells, and this will lead to the interleukin 2 induces expansion of the TC cells clone. And this TC clone started to contact under the influence of interleukin 2, and the expansion under the influence of the contact, start to contact these infected cells with the intracellular organisms. While in case in proteins, the antigen actually is processed as well to a peptide in a phagocytic vacuoles, and as well it is expressed on the surface of the antigen presenting cells, but in this case together with class 2 MHC. Soon after that, this will get recognized by the T helper cell that can bind to this peptide, which is bound to the class 2 MHC complex on the surface of antigen-presenting cells. Soon after that, two major classes of cells or clone of cells, the T helper 1 and T helper 2, arise, in which the T helper 1 will start to secrete interleukin 2. And here you can see how the both branches are related, because you can see that the TC clone can be induced by interleukin 2. And as well, T helper 2 started to be induced in proliferation under the effect of interleukin 2. So let's continue about the branch from the intracellular organism. We end by TC clone contact the infected cells. Soon after contact, the peripherin and the cytokine release occur from these cells, and this will lead eventually to the killing of the microorganisms. This cell-mediated immunity is directly mainly concerned with the intracellular dwelling microorganism or abnormal endogenous cells, even a cancer cell or a virus-infected cell. So in this branch of the immune system, antibody is not involved except in the antibody-dependent cellular cytotoxicity, which is called ADCC, in which the effector cells can be linked to the target cells once it is marked by the antibody, as you can see from the figure. Here you have the target cell. Here you have the target cell, and you can see that the target cells is being labeled by the antibody. And you can see that the effector cells, in this case, the TC cell, can recognize this binding. It's process kind of opsonization. So that's why it's antibody mediated dependent cellular cytotoxicity. So actually, it's not purely due to the effect of the antibody, but here the antibody acts as a bridge between the effector cell and the target cells. And of course, you can see how the effector cells is loaded by cytotoxic granule. And of course, these cytotoxic granule, like the peripherin and cytokine, which will release, especially the peripherin, which will be released over the target cell to kill this abnormal cell, even endogenous cell, even it's a cancer cell. So now let's go back to the other branch. So we have seen that this branch ends by the interleukin-2 secretion from the T helper 1 and the interleukin-2 induced proliferation from the T helper 2 cells. Soon after that, the interleukin-2 secretion induced the secretion of TNF alpha and interferon gamma, which lead to the activation of the macrophage that lead to the secretion of interleukin 12 that promote the proliferation again of more TC and the natural killer cells, which ultimately will lead to the cell-mediated immunity. While the other branch, you can see that the interleukin 4, 5, 10, and 13 start to be released. This will lead, together with the presence of the antigen, this will lead to the B cell activation. And here you can see how the branches of the B cell branch with the T cell branch cooperate. So here, together, these lead to the B cell differentiation and division. Of course, will lead to plasma cell lineage. And of course, you all know that plasma cell that's needed to start to secrete antibodies. Soon after that, this plasma cell will start to do either to secrete IgM antibody or do class switching to form IgG or IgA. In both cases, the secreted antibody can form complex with the antigen. With the antigen, this complex can be phagocytized, and this will lead to something called respiratory burst. And actually, respiratory burst, it's called as well oxidative burst. And this will lead to a rapid release of the reactive oxygen species, superoxide anions, hydrogen peroxide. And if you remember, initially in our initial lectures, we have said that these ROS or reactive oxygen species, one of the warfare of the immune system, one of the weapons of the immune system used to attack foreign antigens. So this will lead, of course, to the lysis of the microorganism. In case of complement, both of them actually, the complement fixation and either phagocytosis ultimately will lead to the lysing of the microorganism. So here, let's see how the antigen processing of the intracellular organism can occur. So you can see the antigen presenting cell can sense the virus. Once the virus is even virus or intracellular parasitic organism, you can see they can internalize inside the antigen presenting cells. Soon after that, in the rough endoplasmic reticulum, you can see that the viral peptide now, of course, if it's a virus, it will generate its own antibody through the process of transcription and translation. Soon after that, and of course, you know that all translated protein are stored initially in the rough endoplasmic reticulum. So you can see here, together with together with MHC, just hold on. Together with the class 1 MHC, you can see that part of the viral protein, of course, through the Golgi complex, because you know one of the major, actually the major function of the Golgi complex is to sort and package and try to release the secretory vesicle toward the cell membrane. So now this viral peptide or any peptide from the intracellular parasitic, which can be processed in the proteosomes, can be expressed on the surface of the antigen-presenting cell. Soon, once it is expressed on the surface of the antigen-presenting cell, this can be recognized by the type of cells that is CD8 T, which actually the CTL or cytotoxic T lymphocyte to initiate the cell-mediated immunity. But in other cases, if it's exogenous protein, So this is actually the diagram that shows the processing of exogenous protein. So you can see this exogenous protein can be ingested either by endocytosis, phagocytosis, or pinocytosis. Anyway, this will lead to acidic endosomal vesicle together with the enzymes will form and the degradation, and together with the class 2 MHC complex, it soon will be form a complex between the peptide of the antigen and the class 2 MHC, which will be recruited to the surface of the membrane of the antigen presenting cells to be, once is exposed on the surface membrane, this will get identified by the T helper cell, and soon after that, this will activate B cell for antigen synthesis. So here, let's talk about the second topic in our lecture today, about the activation of the T cell. Actually, as we said, is initiated when these specific T cell receptor in these kind of cells, either carrying CD4 or CD8, bind to the appropriate antigen-presenting cell peptide MHC complex, either class 1 or class 2. So let's talk first about the activation of CD4 helper, T helper cell, to activate the B cell for antibody synthesis. So let's see more closely and more deeply what's actually is happening. So you see here, if you assume this is the surface of the antigen presenting cells, and the antigen is internalized, it is processed inside the antigen presenting cells and expressed on the surface of the antigen presenting cell together with a class 2. We can see that there is a number of receptors, actually accessory receptor, other receptor that is on the surface of antigen presenting cell, and each of these receptors have a specific role. We have LFA3, we have ICAM1, we have B7.1 and B7.2. Okay, as we all know, this will soon be recognized by the T helper cells. So initially, the T helper cell will recognize the complex between the peptide and the class 2 MHC. And you can see here, you can see that start the binding of the T cell receptor from the membrane of the T helper cell with the peptide on the class 2. This is aided actually by a group of protein which is called CD3, and as well and the famous protein which recognize this kind of cell, the T helper cell, is a CD4. And actually, this complex, this binding between these two group of proteins, this actually leads to activation signal. And this activation signal is transduced over the membrane to lead to the binding of CD2 to LFA3 and the binding of LFA1 to the ICAM1. So why this binding occur? Actually, this binding will add more layer of strength to the binding between the T helper cell and the antigen presenting cell. And what about these two other receptors? Because these two other receptors actually have a major role in the process of regulation. B7.1 bind with CD28, while B7.2 bind with CTLA4. Actually, this binding, the first binding will lead to increase in the senses of interleukin-2. And of course, we have already mentioned a lot of time that interleukin-2, one of the important cytokines that leads to the proliferation of T helper 2, activation of the immune system. What about the other binding? Actually, the other binding leads to the down-regulation of interleukin-2 synthesis. So you can see the mode of the regulation in order to avoid the overactivation of the immune system. Once these binding occur between B7.2 and CTLA4, this will lead to a down-regulation of the synthesis of interleukin-2. And this will regulate how the immune system, and of course will prevent the immune system from being overactive. If we go closely for the other mode of activation concerning the CD8, you can see that it doesn't differ much. Again, the antigen is processed, peptides are expressed together with class 1 MHC, got identified by TCR receptor and the group of protein, which is collectively called CD3, together with the CD8. And of course, you can understand now why it's called CD8, because you can see actually it's 2 copies, if you can assume it like this, 2 copies of CD4, because this one only 1 copy CD4, and here it's CD8. The same receptor, it's the same receptor on the antigen presenting cells, so you can see here, and binds on the cell receptor on the TC cell. And we need to understand why CD22, of course, these binding lead to strengthening of the binding, same as the previous for CD4, but the binding of the CD45R with CD22 again prevent the overactivation of the immune system and development of the autoimmune disease. So it is important requirement for the immune system to regulate itself to avoid any overactivation. So let's talk now about the third topic of this lecture, response to antigen, and we'll talk about the T helper cell response pathway. So actually, as we said, T helper cell actually is classified into 2 compartments or 2 types of cell, T helper 1 and T helper 2. And actually, this actually is based on the cytokine they secrete, which determine the functions, the effect. So the cell induces cell-mediated immunity, following bind, as we said, with class 1 MHC complex together with peptide by transforming, differentiating, and dividing logarithmically and secretes. So if we go closer into 2 helper 1, we can see that it majorly secretes 4 cytokines: interleukin 2, interferon gamma, TNF alpha, and interleukin 12. And of course, interleukin 2, as we said before, it is necessary for T cell and B cell transformation, while interleukin 12, together with interleukin 18 from dendritic cells and macrophage, aid in the transition of macrophage, the movement of the macrophage, the TC cells, the NK cells, and to promote these cells to undergo cell-mediated immunity. While the interferon gamma, it enhances cell-mediated immunity by activating macrophage and NK cells. And as well, it triggers the presentation of the antigen together with the MHC complex by the endothelial cells, and as well suppresses the antibody formation by down-regulating interleukin 4 synthesis. So now we understand that interleukin 4 is very important cytokine in promoting the synthesis of the antibody from plasma cells, while TNF alpha activate macrophage and the synergies with the interleukin 1 in inducing and stimulating, of course, the acute phase responses. All these branches, all these secreting cytokines together with their function, you can understand now it can coordinate the different functions of the immune system, and especially which is related to T helper 1. If you go for T helper 2, actually T helper 2 is more concerned with the activation of B cell and the humoral immunity, following, of course, the binding of the T helper 2 with the peptide bound to class 2 MHC, and of course from the stimulation by interleukin 2, which is secreted by the T helper 1. So it transforms as well, differentiate and divide as well, and secrete. So the T helper 2 actually, as we say, secrete interleukin 4, interleukin 13, interleukin 5, interleukin 10. And of course, interleukin 4, as we have just said, is very essential for the antibody synthesis by the stimulating B cell differentiation, and as well is necessary for the IgE production and the down-regulation of interferon gamma by T helper 1 cells, and thus can suppress cell-mediated immunity. So you can see here, there is a mode of regulation as well between the humoral immunity and the cell-mediated immunity. For the interleukin 10, like interleukin 4, it inhibits T helper 1 release of interferon gamma and the interleukin 2, thus reducing the activation of macrophage by interferon gamma. For interleukin 13, it mimics interleukin 4. So you can see all of these cytokines, actually, their function circulate around the function of interleukin 4. So it mimics the interleukin 4 action. So basically, it inhibits the T helper 1 cytokine release. So you can understand that T helper 2, it's kind of antagonizing the effect of T helper 1. For interleukin 5, it functionally functionate in synergism with interleukin 4 and interleukin 2 to aid B cell differentiation. So together with the stimulation of the growth and differentiation of eosinophil and facilitate the Ig synthesis. So you can see now, as we just said, the T helper 2 mainly concerned by the activation of the B cell and the antibody production. So let's talk now about the B cell response. So actually, as we said, B cell response is initiated by antigen selecting the clone of B cells. That's a very important sentence. The antigen select the clone of B cell that will be differentiated with a specificity that is only specific for that antigen. It's not the B cell select its antigen, okay? So this selection with the membrane-bound IgM antigen receptor that is specific for the antigen epitope. So the binding of the antigen plus the stimuli from the T cell cytokine interleukin 2, interleukin 4, triggers, of course, the division and differentiation of the B cell clone, the specific clone which is specific to the antigen and to the antigen. The interleukin 5 continue this process after interleukin 2 and 4 during the B cell acquire the cytoplasmic machinery necessary for antibody synthesis. Of course, the heavy and light chain are synthesized and assembled under the influence of interleukin 6. So now we can understand the different steps of rearrangement and class switching. Every different step need an influence from a cytokine. This will lead to a terminal differentiation into plasma cell and the start of the secretion of the IgM. The subsequent gene arrangement result in the class switching if the cell needs class switching to IgG, IgA, IgE synthesis and secretion. So to summarize, so interleukin 4 as well, together with interferon gamma, influence the switch to IgG, while interleukin 4 alone influence the switch to IgE. TGF-beta influences the switch to IgA, and the binding of CD40 on the B cell to its ligand, the T helper cell, which is actually CD40L, is necessary for this switching to occur. So to undergo this switching, it needs the specific cytokine, it needs to be in vicinity and bound to the T helper cell. So what about memory cells? So memory cells actually are classes of cells, as we all know, that is generated independent of the plasma cell lineage. And they migrate to various lymphoid tissue, and of course they have extended survival and they offer lifetime, some types of cells can offer lifetime immunity. So what about the secondary response? A secondary response, actually, it is a response to the same antigen. And this, when the body got exposed to the same antigen, actually the body needs a shorter induction period to form the antibody, which is specific, which actually is initiated by the first encounter of this antigen and the memory that the immune system have retained through the first encounter of that antigen. So actually, if there are more rapid class switching, very fast class switching from IgM to IgG, increase the IgG with antibody of higher affinity, even higher affinity than the initial encounter of the antigen. If the antigen is affecting the mucosal tissue, you can see that predominant IgA synthesis in mucosal tissue. And this actually concludes the lecture of the response to antigen.